In this video, we discuss the laboratory synthesis of DNA strands. Many are the reasons why we want to make DNA strands. One of them is to make primers for PCR experiments. Another one is to make small pieces or fragments of oligonucleotides that can be put together to make a gene. The oligonucleotide synthesis is carried out in an automated method very similar to that one that we discussed during peptide synthesis. This is called a solid phase synthesis, and it is carried out on a solid support held between two filters in columns that allow all reagents and solvents to pass through freely. Solid phase synthesis has many advantages. Excess of solutions drive reactions faster, impurities and excess of reagents are washed away, and no purification is required. The goal is to create phosphodiester bond between nucleosides. Synthesizing an oligonucleotide with a particular sequence of nitrogen bases is more difficult than what it is the synthesis of a polypeptide with a specific amino acid sequence. This is because we now have more functional groups that need to be protected and then deprotected at the end of the synthesis. This method will require that the 3' OH be attached to a solid support that we're going to call a linker. Also, the exocyclic nitrogen bases amino groups will be protected by forming an amide bond, and the 5' OH will be protected by forming an ether bond. In the Phosphoroma Diet method, the monomers will be protected on the 5' OH by use of para-dimethoxytrittyl protecting group. The reaction between the 5' OH of the nucleotide will attack the carbon of the DMT in an SN1 mechanism. Before reacting phosphoramidite with a nucleoside, the 5' and DMT protecting group is removed by use of a dilute solution of di- or trichloroacetic acid. In previous chapters, we have discussed the reaction between amines and acyl halides. In these reactions, we have seen that we add a nucleophile to the carbonyl, we create an intermediate tetrahedral, and then we are going to eliminate the weakest base to produce an amide. In similar fashion, we can react the exocyclic primary amino groups of the nitrogen base with acyl halides. Cytosine is protected by reacting the primary amino group with benzoyl chloride, and we see as a product is an amide. Also, benzoyl chloride is used to protect adenine, and guanine is protected by isobutyryl chloride. In all three cases, we have protected the primary amino groups with the formation of an amide. Thymine does not need protection because it does not have a primary amino group. The protected nucleoside is reacted with phenylsuccinate, producing an ester on the 3' of the nucleoside. This succinate of the nucleoside now can react with the amino of the silica gel, producing a linker in a growing chain. When we discussed DNA replication, we mentioned the components necessary, and that included a template. We included activated nucleotides, magnesium 2+, DNA polymerase, and a primer that provided the first 3' OH to start the elongation of the chain growing. I want to emphasize the difference between the nucleotides used in the cell with the nucleotides that we need to create a string of oligonucleotides in the lab. And that has to do with the phosphorylation to be on carbon number 5 for the cell, while in the lab, carbon number 5' will be protected with DMT, and carbon number 3' will be the one attached to a phosphorus. But in this case, we will not have a triphosphate unit, but a phosphoramidite. And what is a phosphoramidite? It is when you have the phosphorus bonded to 2 oxygens and 1 nitrogen. It is only having an octet, while a phosphate will have an expanded octet. In the cell, to create a phosphodiester bond, we will look at the nucleophilic attack from the 3' OH to the innermost phosphorus of the activated nucleotide, a reaction that is catalyzed by DNA polymerase. As a result, the incoming nucleotide forms a phosphodiester bond with the growing chain. The nucleophilic attack of the 3' OH group to the nucleotide leads to the formation of a pentacoordinate phosphorus that leads to the displacement of pyrophosphate. By convention, a polynucleotide sequence is written with the 5' end on the left and the 3' end at the right, in this case, ATG. In the lab, synthesis of one strand of DNA uses phosphoramidites instead of a nucleotide triphosphate. A phosphoramidite phosphorus is bonded to 2 oxygens and 1 nitrogen. The phosphoramidite will react with the free 5' OH on a solid support link nucleoside, here R1, becoming a phosphite. The ultimate goal is to transform a phosphite into a phosphate. The phosphite, this is phosphorus and 3 oxygens bound, is an electron-rich center that can react with water after using iodine as an oxidant. Upon this reaction, phosphorus will have a plus charge. Another oxidant useful for this reaction is 3-butyl hydroperoxide. The unstable phosphite triester is converted to a stable phosphate triester in several steps. Pyridine will deprotonate this intermediate, and at the end of the synthesis of the oligonucleotide, elimination of beta-cyanoethyl group on the phosphate unit yields the natural phosphodiester bond linkage. Let's take a look at the process of coupling step by step. Phosphoramidite will become an activated nucleotide by an acid-base catalysis. Tetrazole acts as an acid. It protonates the amino part of the phosphoramidite. This makes the secondary amine much better leaving group. Also, this will make phosphorus more susceptible to the nucleophilic attack by the free 5' OH of the nitrogen-base protected silica-bound nucleoside. We can see that the electron-rich 5' OH of the nucleoside adds to the protonated phosphoramidite, expelling out diisopropylamine, creating a phosphite. This is 2 units of deoxyriboses linked by phosphorus on carbon 3' and 5'. I am using the picture from your textbook to summarize the process. A phosphoramidite is activated by the catalyst tetrazole, and it produces a phosphite. But we need to transform a phosphite into a phosphate. This will take place by oxidation of phosphorus while keeping all groups protected. Only DMT on the growing chain will be removed to allow the synthesizer to start all over a second cycle. This process is called detrylation, which I will discuss later on. This is a summary of the requirements for the oxidation. The phosphite gets oxidized by aqueous iodine in the presence of pyridine. We will work the mechanism of this step. Electron-rich phosphorus reacts with iodine, spelling out iodide ion. At this point, phosphorus has not expanded its octet. Therefore, similar to the chemistry of nitrogen, it acquires a plus charge. This cation is now susceptible to bond a molecule of water. In this way, we now get the fourth oxygen needed to make a phosphate. This intermediate is very unstable. Proton abstraction by pyridine will expand the octet on phosphorus, which is now transformed into a phosphate. Now we're going to explore the mechanism of DIT relation. This is an SN1 mechanism. A weak diluted acid is needed to prevent the protection of the nitrogen bases in the growing chain before completion of the synthesis of the entire oligonucleotide. The ether group on carbon number 5' is protonated by the weak acid. Once carbon 5' oxygen is protonated, the presence of two methoxy electrodonating groups triggers the release of the nucleoside. We can push electrons and see that the product is a very stable carbocation plus a free nucleoside. This byproduct is very stable due to the three contributors resonance structures. Ammonia will be used to deprotect all nitrogen bases and the cyanoethyl group at the end of the synthesis. The protecting groups must be removed so that proper hydrogen bonds between oligonucleotide and the target nucleic acid may form. For example, once the nitrogen bases are deprotected, a primer made in this fashion will anneal to a template during a PCR experiment. The deprotection of the nitrogen bases is a typical reaction of an amide with ammonia. We add the nitrogen to the carbonyl of the amide group. We create a tetrahedral. Then, after a proton shift from ammonium ion, the tetrahedral collapses, reforming the carbonyl. This will kick off benzamide and the unprotected nitrogen base. To form the phosphate natural form, as in DNA, the beta-cyanoethyl group of the free oxygen of the phosphate is also removed by ammonia. The process is a beta elimination. If we recall from chapter number 22, the chemistry of the alpha carbon, the alpha carbon of nitriles is weakly acidic. You can see here the pKa is about 25, that is without the phosphate unit neighboring, which is comparable to the pKa's of aldehydes and ketones. During the beta elimination of the beta-cyanoethyl group, ammonia deprotonates the highly acidic hydrogen on the alpha carbon, which leads to the expelling of the phosphate unit. This forms the phosphodiester bond between the two deoxyriboses. The byproducts are acrylonitrile and ammonia. Recall that the protecting groups of phosphate and the nitrogen bases are removed in a single step with the aqueous ammonia. The process repeats, and this is an efficient method of addition step by step that allows the addition of 70 to 80 nucleotides. DNA synthesis facilitates the synthesis of primers for PCR, is useful for gene therapy and other methods. Thank you for watching.